And now it is my pleasure to introduce Giorgio Scita from IFOM and University of Milan, who is a biologist. So this is the first talk from a true biologist at this meeting. Today you will get two, because there is also Diana there in the audience. All right. And so yesterday he suffered a bit with the second part with a lot of physics and equations. Yeah. Today you have your occasion to make them suffer.
Yes, I have my revenge, definitely.
Get your revenge.
You definitely get my revenge. So the disclosure is actually done, and what I'm going to be telling you today is a work that we have been doing in the past, well, actually almost 10 years, not by ourselves, on the notion of tissue fluidification or solid to liquid-like transition. I'm not sure what I can say jamming-like here, in pathophysiology, and in particularly I will dwell on a set of unpublished data that through which we use contact percolation theory to interpret some of the heterogeneous mechanical property of tumor tissues. So this could have not been done, and if you have specific questions on the physics, you have the guy to ask them, Fabio Giavazzi and Chiara and Roberto. So there has been a long-standing collaboration. And the work that I'm published that I'm going to be telling you about is carried out primarily by Leonardo and Camillo Mazzella in the lab. All right. So we have been interested in tissue fluidification, tissue transition in the context of cell biology, because like ice cube that melts, even spheroids that actually are prone to become fluid or have fluid-like property may actually increase their ability to spread when fluid diffusion is increased. Very much like an ice cube that is melting. Increasing fluidity enables this spheroid to spread at a much quicker pace. So this belongs to a general framework where phase transition have been studied in the context of biology because they impact on a number of fundamental biological processes. And during, if you think, organ development, any organ would tend to develop into a solid, dense masses which allow them to acquire their rigidity property, their elasticity property, as well as their barrier property. However, if you need to remodel these tissues or undergo morphogenesis, like in this work by Diana Pinheiro, that I guess is going to be talking. Ah, there. Ciao. Yeah. And C.P. Eisenberg, you will need a certain degree of fluidification to take place to allow morphogenetic processes to occur, and she beautifully shows a graded solid to liquid-like transition driven by increased cell-cell motility. We have also looked in adult tissues, and we have been particularly focused on tumor tissue-like, where you have, and I'll tell you how we do that, a control and a more fluid-like tissue, which is confronted with narrow gap passages, which are the same size of the spaces and channels tumors will encounter when invade into the surrounding stromal matrix. And if you're fluid, you are much faster in going through this narrow gap. Even in 3D, if you have acquisition of fluidity, that translates in a persistent rotational motion. What you're seeing here is a spheroid, again, embedded in thick collagen matrix, which is interspersed with fusel GFP marker, which we have exploited in the past to actually study how the rotational motion of this entity exert traction forces sufficient to modify the extracellular matrix. So all these processes can be viewed for the length of Max is here. And so he's one of the first that proposed this notion of jamming transition, which is a phase transition characterized by highly coordinated collective motion and very much similar to the motional ensemble of living beings. All right, this set the stage. Where do we study this? We study in a pathology. I'm a cell biologist. I'm a professor of pathology at the University of Milan. And so the pathology we are looking at, it's ductal carcinoma evolution. And an early type of lesion in the spectrum of ductal carcinoma, of breast carcinoma, is defined as ductal carcinoma in situ. You know, biologists love acronyms. And DCIS, which stands ductal carcinoma in situ, tells you that this carcinoma typically develop within the mammary duct. They are confined, encased by the myepithelial layer. They grow, and so frequently they reach a high packing density, which is typical situation where you would expect the lesion to become solid, inert, sessile. And indeed, 70% of this guy, and I just want to point out from the pathologists that are not here, but it's here, that 20% of all breast cancer diagnoses are DCIS. And we have no way to detect and to predict the one that actually will invade from the one that will stay indolent. And they might stay indolent because, as I said, they are solid-like and inert, and this exert a sort of like tumor suppressive mechanism. And in fact, 70% of all these DCIS will not be needed to be treated. But we have no way to discriminate, so all the patients are treated. And we would predict and expect that in order for this solid-like mass to become invasive, they would acquire a certain degree of fluidification. All right, within this context, the best way a cell biologist can look at fluidification is looking at the model monolayer. That's what you see here. This is doesn't really matter the cell type, is a normal mammary epithelial. This is covered the entire space, as you see, as a tile, and tessellate the entire space of view. And then we look at the dynamic evolution as the cell keep on dividing. And at the beginning, the system is virtually fluid. So there's a lot of local cell rearrangement between cells. As the density increases at a particular shape transition, maybe Max will tell you more about this, there is a transition to a solid, rigid-like state, which can be captured by many different ways. Fabio and Roberto actually devised particle image velocimetric analysis to define the vector from this phase transition, from these phase contrast movies, and you can follow the evolution of the velocity of the vector velocity over time. You can actually depict the alignment factors and which capture the emergence of a dynamic heterogeneity, because in yellow you have cells that move with a perfect aligned average field, vector field, which is similar to the global vector field. In blue, you have opposite moving cells. Within this context, fantastic work by Fredberg, by Xavi Trepat, that is going to be coming tomorrow, yes, by Max, Tommy Angelini, and before, Peter Claudin and Sidney Nagel, actually provided a framework to try to interpret this transition in the context of living matters, obviously. And the beauty of this framework, this jamming, solid-like, to unjamming, fluid-like transition, is that it is dictated by a set of simple biomechanical parameters, some of which are very obvious. Actually, the strength of cell-cell adhesion, which is actually less obvious from a cell biological point of view, because always a tug-of-war between the strength of cell interaction and the atomizing contractility that tend to break this interaction. Motility, I know you discuss a lot about an effective temperature of system that stays at the same temperature on Monday. I wish I could have been here. I missed this discussion. But cell motility is a sort of like intrinsic activity of individual cells that may actually switch the system from a solid to a fluid-like state. And other that are a little bit less obvious, like cell shape and shape variance, that have been associated invariably to a state of fluid versus solid-like being hexagonal, pentagonal, having a defined minimized state of geometric energy, if you wish. And why we are interested as a cell biologist and cell pathologist to this, because this is a framework which is an alternative gateway, if you wish, to cell migration. Even if you're a physicist, I'm sure you're familiar with epithelial to mesenchymal transition. So an epithelial carcinoma, to acquire migratory property, need to become something different from itself. We need to acquire capacity of migrating, of polarizing. And in this case, this transition pertains to the entire system and is an emerging property of a collective system, and simple changes in biomechanical property may switch from a solid to a liquid-like state. For this length, you can revise some early work. This is beautiful work, 2 Nature papers, that actually were the first to propose early dissemination during breast cancer development, during carcinoma development. And what they show is that this lesion, which are ERBB2 and oncogene in breast cancer, that make the tumor very aggressive, at this stage, when the mass are dense and compact, are much less prone to metastasize with respect to early lesions that are presumably more fluid-like. So this is why we are very interested. That's why as biology, we are very interested. And of course, as a biologist, you ask basic fundamental questions in terms of molecular details. And so we ask how the cell control this transition, what are the immediate pathophysiological consequences of it, and we dwell in the last couple of years, more, in studying what are the long-term consequences. I'll provide example of all this and dwelling more on unexpected link. Yeah.
I just want to make sure I understand which phase transition you're talking about.
All right. Well, we are.
So, I have a phase diagram. There are all kinds of boundaries here.
Yeah.
Are we talking about near a
something
critical?
that is solid and that can be switched to become fluid in many different ways.
Through which process? In the different
Many different ways.
Okay.
In many different ways.
Are we near a critical point or near a phase?
There are critical points, and you need to overcome critical points in order to become.
Well, which critical point are we talking about?
It is dictated by the boundaries.
Okay.
And so any perturbation of these features may actually switch a state, an epithelial collective state from a solid, defined solid as in a very simple term, which is the one that I understand. Individual cell can no longer rearrange.
Yeah, I just don't know if all of those lines are the same kind of critical phenomena.
Yeah.
So I was just
Yeah, this is
wondering.
maybe Max can actually also interject and discuss this. All right. All right, we started with the first question. How the cell control this phase transition? And here I need to spend a couple, my revenge is here. Because we focus on this guy, which is ROD5. The only thing you need to know, it's a gene product. Actually, it's a family of genes, but only one does the trick. That is absolutely essential to generate this early endosome. Early endosome are structures on which anything that gets internalized ends up. And here you decide whether the cargo that comes from the plasma membrane or lipids recycle back or need to be degraded. So it's in biology, we call it sorting station. These are decisions that needs to be taken. In absence of ROD5 or perturbation of ROD5 level, alter this process, and it's going to affect a number of different cell biological processes. Why we focus on this? Because if we look in breast carcinoma, in human breast carcinoma, and we look at the expression of ROD5A, we invariably see that elevated tumors with elevated expression of ROD5A perform much poorly. They have a decreased disease-free probability, particularly when we look at a particular subtype of breast cancer, which is very difficult to predict in their outcome. All right, so this gene, when we plated our model monolayer in a dense, solid jam state, and we simply induce the elevation of the expression of this gene to mimic the situation that we see in tumors, you see something amazing, because the simple expression of a single endocytic gene transforms this immobile, rigid-like tissue into a,
How is the expression is triggered? Like it's the
No, the expression is due to a lentiviral infection of the cells that is controlled by doxycycline. So we are doxycycline, we control the expression.
Then how long after doxycycline?
It takes 6 hours to see the expression of the protein, and then if you keep doxycycline, the media will stay high. All the movie that you've seen, that we will see, are movie in which we add doxycycline. We start the monitoring by time lapse, and then we follow it for 24 to 48 hours. It takes about 10 hours to reawaken the cell locomotion, and this coinciding with the expression of the protein that takes initial expression of, and then it will done. I'll show you in a second. Yeah.
Slide and explain a bit more what is this protein doing? I mean,
So this protein, what it does a couple of different things on endosome. So endosome, I hope I don't need to explain too much on it, but are organelles that receive cargoes from the plasma membrane. So you have a constant trafficking or cargo or plasma or lipids from the plasma membrane to the inside of the cells. And so the first point where this cargo ends are early endosomes. Number 5 is necessary for their biogenesis, for their fusion, and for their dynamics. And so and not only that, it's also necessary because then the process becomes more complex. The early endosomes have to mature, acquire other additional markers, which enables them to either act as a recycling endosome or as lysosome, which are degradative organelles.
Is transporting the cargo, or it's affecting how the endosome is working?
It's affecting how this endosome function, the size, and the motility. It doesn't affect the internalization cargo.
If you change this expression of this protein, you change how the endosome works. So
Exactly.
you do something many things in the cell.
Exactly, that's exactly the point. However, there is specificity because there are 3 genes that encode for this guy. Only one of them does it. The other that, if you look at the sequence, are basically identical, don't do it. We can discuss as to why is that. But it's true, that's exactly the point. You're gonna be affecting a number of different biological processes. If you think about junction, for instance, junction is constantly to be internalized and recycled. If you think about receptor tyrosine kinase receptor that respond to EGF, they constantly get activated, but as they get activated, they get internalized, recycled. So you're going to affect a number of different processes, inevitably. All right. So this is the phenotype we are looking at. So if you look to answer the previous question at a number of different parameters, these are simple ones like root mean square velocity. As you increase the level of expression of phi, you have a peak of motility that then subside. We can discuss as to why that is the case. I can tell you why. It doesn't have to do with the level of expression of phi. It has to do with the factor, which is EGF, epidermal growth factor, that is essential for this motility to take place. And what RAB5 elevated expression does is to reduce the number of receptors on the plasma membrane. All right. Then you can look at the correlation function, the velocity vector, extract length of correlation to get a quantitative view of the size of pack of cells that move in a coordinated fashion. You can look at mean square displacement over delta t, which indicate that these cells or this group of cells actually move in a ballistic fashion. And Roberto and Fabio actually made sense of some of these observations by doing a very, I think, at least up to my eyes, was a very clever experiment, was to look at the motility of this monolayer from a reference frame that is either the cells or the entire monolayer. And so if you do that by basically removing the translocation, you see local fluctuation of cell velocity, fluid-like type of behavior, I would say. If you look at the reference frame of the investigator, then you start to see this flocking emerging. They also did, all right, so this is very similar to what we had seen in Milan. This is a sterinite, the moving sterinite, where you might have what I would define as solid flock, but then we can discuss that in a second. Because then, of course, they use a model. Again, the detail you can ask them. It's a self-propelled Voronoi model, vertex model, where there are 2 major ingredients. One is the target shape, the shape of each individual cells, perimeter versus square root of the area. The other is an alignment interaction, J. And so this is actually taken from Roberto. So if you have a solid-like below the target shape index, you have a solid type of tissue at low alignment interaction. If you increase the target shape, you start to become and acquire local cell rearrangement. And then you might have a situation where you have solid flock, which we had never seen until a few months ago. Because then we were collaborating with Johanni Vasca, looking at the motion of a type of cancer which grows in your vocal cords. And this type of cancer acquires solid flock type of motion naturally due to a number of oncogenic perturbations that this cancer are subjecting to. And then what we were interested in is this situation where we increase the shape index, they increase the alignment interaction, and then you start seeing what we define as solid flow, as liquid flow.
Showing fluidization and when it's not.
Yeah.
Yeah, I can't buy. Yeah, can you explain a little bit what is changing? Obviously, something is changing in the.
All right. Here, the only thing that has changed is target shape, the shape of the cell, right? And under condition in which that's the vertex model that I know, Max, you're going to be talking about more in detail. And the major feature, I mean, there are equations that of the energy of the vertex, and then the only thing really that you change, that the critical parameter is the shape. All right. All right. Even more striking is what happened in 3D, because now we have variants, oncogenic variants of these normal epithelia that I've shown you before that actually can form spheroid or tumoroid. And then you can do the same trick. What you see here is the M-cherry H2B label to allow the visualization of each individual nuclei. If I turn on ROB5, then you start to see this persistent rotational motion that lasts a couple of days, 3 days sometimes. We have actually looked, Fabio has looked, to see whether in addition to this global coordinated translocation, you have also local cell rearrangement. You do, and is graded from the center to the periphery, meaning that the periphery is sort of melting, enabling local cell rearrangement, particularly at the periphery. And why is that important? Because these concur to determine a number of different features. Because now if we embed these guys into a thick collagen matrix to mimic the stromal desmoplastic reaction that is typically observed in breast tumorigenesis, in carcinoma in general, what you see that at first they start to rotate, they exert large traction forces that Fabio and Roberto could calculate that are sufficiently high to modify the extracellular matrix and to generate gap or individual channels into which these cells actually invade locally. This is not just an oddity of an in vitro experiment, because if we look in human DCIS, they typically express low level of ROB5, which become particularly prominent, and I come back to it in a second part, at the site where local invasion, and I would argue fluidization, takes place. And so the model was that flocking fluid motility through a set of mechanical perturbation enabled the escape from a jamming tumor suppressive state. All right, take a breath. And now it's more biology. Well, not only. Because we ask, I told you the analogy between epithelial to mesenchymal transition. To become mesenchymal, you need to change to rewire your transcriptional profile, your identity. And so there are implications in becoming mesenchymal that goes beyond the ability to move, but the acquisition of traits that enables those cells to seed metastatic foci. So we ask whether under condition of solid to fluid, we would see something similar. And I summarized everything in one single slide. And we show that as the tumoral tissue that undergo a solid to fluid-like transition, which enables them to invade locally, they have to pay a price. And the price they pay is that they're undergoing tremendous fluctuation in their shape, and their strain is significantly increased, which is translated, in my view, is that they are subjected to persistent mechanical cyclic stresses. These are transferred to the nuclei, and we can actually provide direct evidence that nuclei undergo large strain, and this, in turn, through a set of mechanisms that I'm not going to have time to tell you about, eventually lead to rupture of the nuclear envelope. And rupture of the nuclear envelope is a problem in biology because, well, we have just come out of COVID. Infection, viral infection, which put a lot of oligonucleotide in the cells, is sensed by an ancient innate immunity pathway. This SIGAS has a very high affinity for cytoplasmic DNA, gets activated and turns what? Interferon. Why? Interferon block viral replication. They block the replication of the cell in which the virus is entered. So it's a very ancient evolutionary pathway that eventually lead to the boosting, the dramatic boosting. We were totally surprised of this because we get 1,000-fold upregulation of an inflammatory gene signature. So the change in identity is due to this hyperactivation of SIGAS-STING that eventually leads to a dramatic inflammatory response, which has dramatic consequence because it comes with the acquisition of traits that are essential for seeding distal metastasis. I'm not going to show this to you today. This happened in real tumors because if we again, these are human DCIS. In green, you see RAP5, which is typically upregulated at the site where eventually this tumor becomes invasive, along with CGAS. This is the sensor, the innate immune sensor, in real tumor samples. The other thing you can notice here is that this tissue is heterogeneous. So you have local variable expression of RAP5, which is invariably accompanied also by a change in cell shape, in target shape, if you not in target, in cell shape, suggesting that maybe what we are seeing here is not only a transcriptional heterogeneous distribution of an essential gene, but also an heterogeneous, a mechanical heterogeneous state, which we have not mimicked in our previous experiments. So we wanted to address the question as to whether this mechanical and transcriptional heterogeneity impact on the dynamic and breast cancer, on the dynamic of the tissue, on the breast cancer progression and the genetic rewiring of cells. So to answer this question, actually, this is an experiment that we had done a few years ago by Chiara Malinverno that was resumed by Leonardo, where we actually take advantage of the fact that we can have stable cell line that can be used to express the RAP5 gene that are labeled. Their nuclei is labeled with EGFP in green or mCherry in red. And so you can distinguish the two cell type. And then you do a very simple experiment. You mix them and you generate monolayer with a different fraction composition of the fluidized cell. And so this is the first, actually, still the best movie that we got, where you go from 100% control, all green, 100% RAP5, all red, and then in between you have various fraction composition of the monolayer from 80 to 20, 70 to 30, 50 to 50, and so on and so forth. And then if you run the movie, you see that you go from a solid to a flocking fluid, and then in between you have a range of global acquisition of cell locomotion. Yeah, all right. All right, so you can actually determine what's going on by plotting, for instance, the root mean square velocity. Well, as you increase the fraction of RAP5 cells, everything moves more. What is more striking is when you plot parameters that are indicative of flocking fluid, of flocking emergence, like this polar order parameter, which goes from 1 to 0, 1 when they're all aligned, and then and 0 when they are totally move in a totally uncorrelated manner. And then you have a threshold-like behavior. Above a critical threshold, which is above 50%, you start to see flocking of the entire system of the control that are immobile and there are 5 cells. All right, that set the stage. And then Fabio actually was inspired, I guess. I don't know why, how, but he started looking at the composition of this monolayer as you increase the packing fraction. Nothing really fancy, because you would expect if you change the composition that at a certain stage you might actually generate a cluster that is spanning the entire field of view, a percolating cluster. And so the question, and it is remarkable that, not remarkable, but this is a geometric inevitable outcome of this situation, that above 50% you have increased formation of percolating cluster. And he actually did that, and I go quickly, by taking the centroid of each cells, defining the Voronoi segmentation, and then defining the boundaries between each individual cell that are either red, expressing RAP5, or green, expressing control cells. And you can, we can actually observe that the percolation transition actually coincide perfectly with the flocking-like transition. All right, this is just correlation though. We wanted to have some indication that might be these two processes causally related. And to do that, one experiment that we did was to try to generate a model monolayer where the total global situation is above the 50% threshold, but then you actually interrupt the formation of percolating cluster by plotting 100% RAP5 here and 50-50% control and RAP5 here. And then you don't see the entire system to flock. You just see local flocking, if you wish. You can quantify this by plotting the local decorrelation rate, indicative of the fact that you might need and is essential to generate a flocking, a percolating cluster for flocking to emerge. Then again, thanks to him, he started to play around with some models. And the simple model that he had was actually looking at the cells as disks, soft disks, with a certain prescribed ability to realign the locomotion. In control, they are randomly rotating. In RAP5, they are linear. And then if you plot as a function of the aligning cells and you look at the motility of this guy, you realize that it's enough, a small percentage of non-aligner, of dissenter, as we call it, to disrupt the flocking fluid type of motion, which is not what we see. All right, so are control really dissenting then? Are they stable in their phenotype? And we actually hypothesize that in function of the surrounding of the control, you actually change the dynamic shape, junctional property of the control cells. So to prove this, we actually first turn to into model. And so if you mix aligner and non-aligner that can become aligner, then you start to recover a threshold-like type of behavior. But of course, we are biologists. We are not satisfied with that. We want to know the molecular detail that may underline this process. And we set out to do 3 sets of experiments. One is to look cryptic lamellipodia. This is a oddity of cell biologists. But if you are in a dense monolayer, if you want to push and move, the only way a cell has is to extend the protrusion underneath its neighbor. These are what are defined cryptic lamellipodia. And that can be used as a parameter of polarity, if you wish, or local alignment. We are looking at cell shape, and then I end up with gene expression in a second. All right, to look at cryptic lamellipodia, there is a beautiful experiment that one can do. You can express mosaically life-like GFP, and here we are looking at 100% control or 100% RAP5. This GFP, of course, they express either control or RAP5, and then you can, I run the movie because it's easier, and then you can plot the local velocity vector with respect to the orientation of the cryptic lamellipodia to understand whether these cells align in the direction of the local motion. And now we do the same experiment in, again, 100% control. They are randomly oriented. In RAP5, we knew from previous data, they are oriented toward the local velocity field. And then if you do under percolation, above percolation, under the threshold of flocking fluid motility, you still have a random orientation. If you are above, you start also the control, start to realign and extend protrusion along the local direction of velocity. The opposite is also true, because now if you are under the percolation threshold, the RAP5 lose the ability to extend this protrusive activity in an oriented local manner. All right? The other experiment you can do as a biologist, you can perturb the ability of control cell to extend this protrusion. So you make the dissenter more dissenter. And then you can use this trait by expressing a dominant negative rho GTPase, which is RAC1, which is essential for the formation of this protrusion. And now you start losing the threshold. All right, and then I quickly, because I'm running out of time, if we look at shape, you can see that 100% control are more like close to the 3.81 perimeter versus square root of the area. The 100% RAP5 are elongated. And in between, you start to see that when you are below the percolation threshold, the RAP5 become more pentagonal, hexagonal. The opposite is true for the control. And you can quantify this, and you can see that there is an increase in target shape as a function of the fraction of RAP5 expressed in cell, particularly above the percolation. Finally, and then I shut up, the other experiment we can do is the following. Since the cells are labeled fluorescently, we can mix them, recover them by FACS sorting, and ask the question whether the cells that have acquired this flocking fluid motility, the control in particular, also acquire traits that are transcriptional in nature. All right? And of course, we did RNA-seq, and I'll just do this because it's easier. What you see here, is a core set of these interferon SIGAS-dependent genes, a bunch of them that you see are upregulated dramatically in RAP5 100%, but they're also upregulated in control cells, this is in control cells that are intermixed with RAP5 above the percolation threshold. And so you can plot this, the log2 normalized counts of all these genes, which indicates that above the threshold of percolation, also the control become able to transcriptional rewire their state to a more RAP5-like situation. All right, let's say I can skip this. So just to summarize what we have seen, then we can discuss. Below the percolation threshold, you have a situation where increased number of RAP5 actually increase global root mean square velocity. Above the percolation threshold, you start seeing the emergence of flocking fluid motility, the acquisition of an interferon stimulating gene signature, which sort of make even the control more similar to the highly invasive oncogenic cells. And so there is a phenotypic switch of control as a function of the surrounding cells, characterized by upregulation of this interferon-regulated gene, active extension-oriented pertusion, and elongation of cell shape. Now, what we are doing, I'm not going to show anything, I'm done, but we are studying what is the nature of this intercellular communication as a function of the RAP5, which is, I anticipate to you, chemomechanical in nature. They are both factors that are released and influence the control, as well as it is necessary to have contact and a local surrounding in order for control to change their transcriptional state. All right, and this is all right, I'm done. So I already mentioned the people in the work. This is the group. I have to thank the various agencies actually that supported our work. And thank you a lot for your attention.